Okay. First of all, I would like to thank the organizing team for having me here and giving me the opportunity to present some of my research. I will straight away go into the presentation. Okay. Now, fish populations in the wild are exposed to or confronted with various kinds of predators. This could be natural predators like the fish predators or the birds, and this could be artificial or human-induced predators. When I'm talking about artificial or human-induced predators, I'm referring to the fishing gears. Most fishing gears across commercial or recreational fisheries tend to harvest, that is preferentially catch and take out of the populations large-sized fish. Now, when this happens over many generations, it leads to a selection for small body size. On the other hand, there could be gate-limited fish predators which specifically catch small-sized fish, sparing the large ones. There could also be certain kinds of fishing gears which have size limitations, which specifically catch small-sized ones, again sparing the large ones. So a selection for small body size or large body size will ultimately lead to a change in life history traits. For example, when there is a selection for small body size, this would lead to the evolution of a fast life history which is characterized by early maturation at small body sizes, increased reproductive investment, and reduced terminal body length, which means that the current reproduction is prioritized over future reproduction. In case of a selection for large body size, it would lead to an evolution of slow life history, where future reproduction is prioritized over current reproduction. Now, let's assume, assuming that the amount of energy that is available within an organism's body is constant, so if there is more investment in one kind of an expensive tissue, structure, or bodily process, this could be traded off with reduced energy investment into another kind of expensive tissue or process. Now, reproduction is energetically expensive, which means that development and maintenance of reproductive structures and processes is energetically expensive. So a higher energy investment into reproduction could be traded off with lower energy investment into other expensive tissues. We know that the most expensive tissue structure or organ in the body is the brain. So we can expect that there would be a trade-off between energy investment into these two expensive structures and processes, that is reproduction and brain. So in case of evolution of fast life history, which we see in case of evolution of a small body size, we would see that we can expect that an increased reproductive investment, energy investment into reproduction, would lead to a reduced energy investment into the brain, and that can translate to reduced brain size and changed neuronal packaging in terms of, that is, reduced number of neurons and density of neurons. We can expect just the opposite pattern in case of slow life history evolution, where the brain size could be increased and the neuronal packaging could also be simultaneously altered. Now, as brain is a seat of cognitive abilities, changes in the brain would lead to altered cognitive abilities. Now, this can trigger a lot of have a lot of consequences. It can trigger a chain of events, not only at the individual level but at the collective level, population level, and ultimately at the ecosystem level. We put forward this as a novel hypothesis which has been recently published. If you are interested, you can please take a look at this paper. So, going to the questions that we ask in this study, we ask two principal questions. First, does size selection alter brain size and brain morphology? And does size selection alters individual and collective cognitive abilities? So the hypothesis is that if there is a higher reproductive investment, this would be traded off with low investment in the brain, as in case of evolution of fast life history. So the brain size would be reduced, or morphology will be altered, and this would lead to reduced individual and collective cognitive abilities. Okay, now the experimental model. We used an experimental evolution approach using zebrafish as a model system. We intensively harvested wild populations of zebrafish to generate 3 selection lines. The small line was generated by removal of 75% of the largest individuals out of the population. The large line was generated by removal of 75% of the smallest individuals, and the control line was generated by removal of 75% of random individuals. The small line is basically the interest line because it's kind of the fishery line. We have 2 replicates for each particular selection line, and we conducted the selection for 5 consecutive generations. A bit of a background about the selection lines regarding the divergences in life history and the behavioral traits. So as expected, the small line which evolved a fast life history showed early maturation at smaller body sizes, had increased reproductive investment, and also evolved a smaller terminal body length compared to the control line. They also showed divergences in behavior. So the small line showed shy behavioral tendencies, while the large line was consistently bold across different ontogenetic stages. The small line fish also formed less cohesive shoals, while the large line formed more cohesive shoals compared to the controls. Okay, the first question. We ask how size selection alters brain size. This is a work done by Ho-Chi Othniel Wong, who is a master student who worked with me in this project. This is how a zebrafish brain looks after dissection. So we sampled 240 fish, 40 fish per selection line. We sampled them at 290 days post-fertilization age. We euthanized them, measured the standard length, measured the, we dissected the brains out, we photographed and measured the different brain regions. And we used ImageJ for analyzing the size of the different brain regions. So this is, these are the different measurements that were conducted. We measured the sizes of the two tencephalons, the optic tectum, cerebellum, medulla oblongata, and the two hypothalamus. We use this formula to calculate the brain volume, and we used brain volume as a proxy for brain investment. So what did we see? We see that when we compared the brain sizes across the different selection lines, as you can see in the plot where we plot the brain volumes across standard length, we saw that the large line fish actually evolved significantly larger brains compared to the control, but we did not see this effect in case of the small line fish. So this kind of partially agrees with the energy expenditure hypothesis that I talked about before. We also saw changes in the brain morphology. Large line fish evolved larger cerebellum size, while the small line fish had smaller medulla oblongata compared to the control line. Now we move on to the testing cognitive abilities in individuals and collectives. So we tested individuals in inhibitory learning. So inhibitory learning is a kind of an executive function where the organism has to inhibit its unresponsive behavior or reflex behavior when it does not achieve a goal, and stop doing that and do something else in order to get to the reward or get to the goal. So we used a detour test to assay this. This is a classic test used across different species of animals. So if you follow the first setup on the left side, this is the habituation phase where we placed food outside in the arena, and there is a transparent cylinder which is opened from both ends. So the fish has to come in close contact with the cylinder, and once it does, it gets the food. After the habituation phase, we subject these fish to 10 test trials where when we put the food inside the transparent cylinder. Now, here comes the reflexive action. So the fish have a reflex behavior to constantly nibble at the transparent surface in order to get to the food. So it has to learn to stop doing that and instead take a detour through either the right or the left side and get into the cylinder and grab the food. So we subjected the fish to 10 consecutive trials, and we scored the number of successful detours after the fish were released and the number of redundant attempts to access food once they were released. What did we see? So we compared these two estimates across the 3 selection lines. We would expect that if the fish had set inhibitory learning, the number of successful detours would go up while the number of redundant attempts would go down across trials. We actually found opposite patterns in the small line. So the small line fish actually showed reduced inhibitory learning abilities compared to the control line. We did a collective learning assay. We used an associative learning paradigm for doing this. It's a color discrimination paradigm where the fish learns to associate one of the colors with the food reward. We did this across ontogeny. We started with juveniles, then subadults and adults. We subjected groups of 8 fish to this particular assay. We released them into the arena, and then when a random individual entered the correct colored chamber, it was rewarded with food. We estimated the time taken by this random individual to reach to the food reward and the number of mistakes made by the individual before it reaches the food reward. We plotted these across, we compared these estimates across different selection lines. We again saw that the small line fish showed reduced collective associative learning ability, both as subadults and adults. So if you see the graphs that follow, and if you follow the red lines, so the time taken to reach to the food reward and the number of mistakes made actually does not change compared to the control line for subadults, and it actually increases, whereas it should decrease if the fish would have learned for the adults. So coming to the conclusions, we saw that size selection changes brain size and brain morphology. How? So we find partial hypothesis for the energy expenditure hypothesis because we found these effects only in the large line and not in the small line as expected, against what we expected actually. We found larger cerebellum in the large line fish, and this could facilitate better motor controls. The large lines were always bolder and faster in reaching to the food rewards, and probably this could be the reason why, larger cerebellum could be the reason behind this. Smaller medulla could explain lack of flexibility in changing the behavior, which we see in case of reduced inhibitory learning abilities in the inhibitory learning assay for these fish. So there could be another reason, and this is something which we are doing right now. We are estimating the number of neurons and the density of neurons in the different brain regions and the brain overall. That could be better explain these results probably. So we are doing that, and there is also a lot more to this story. With this, I end my presentation. I would like to thank my core team, Hobart, Pavel and Valentin, and my collaborators and the different funding agencies, my host institute, IGB Berlin, my research group, Social Ecological Fisheries Research, the funding agency, the German Research Foundation, and all the partner institutions of the Berlin University Alliance. Thank you very much.